because we were already working on influenza, it was quite timely to switch to doing some work on COVID-19. So that became the bigger part of this talk. So if you want to do some modeling, a lot of people go to our website, glycam.org, and when you land here, it's not particularly obvious, if I'm honest, it's not particularly obvious what you can do. We have tools down here that talk about things like building a glycoprotein or building a carbohydrate. Well, these are phrases we use, but they don't necessarily translate to new users. And things like glyspec grafting, good Lord, what does that mean? So we decided, and we got funding to create a very different sort of interface. And the goal here is to create a suite of tools that are appropriate for non-specialists. And the reason we're doing this is because if you think of people who are able to model carbohydrates, currently most of them are really specialists. They have to know glyco and they have to know computational stuff. And what you really want to translate tools into a broader community, into, for example, the mainstream computing community that doesn't know anything about sugars, or the mainstream glycobiology community who aren't specialists in computing, is you need a different way to interact with people. So my model is something like Geico, right? Where it's, you don't have to understand insurance to be able to get what you need. So we decided that what we would do is we'd make some approximations, so we don't give every option to every user, but keep the idea that you have to make as few clicks as possible to get a useful answer. And the idea is really that this is what's necessary if you're going to translate modeling into the hands of people who can use it as a tool to advance their science. And this is what it looks like. This is now the development stage, so it's dev.glycam.org. And dev means it's prone to change, right? It's not mainstream, it's not permanently stable, but if you want to experiment, this is where to go. As I mentioned, it's for non-specialists. It's, as you can read, what we've adopted is as an interrogation-based interface. So when you go here, you just naturally read, what are the shapes of my glycan? Oh, I want to know that, so that would be the tool. Or search the PDB for glycans. Okay. Or check the quality of my structure. Or how does glycosylation affect my protein? By approaching it this way, because these are questions we've all in the field been asked, then users have an idea of what they're going to get when they go there. And what I'm going to start with, before I get to the COVID work, is the tool that I was originally going to discuss, which is searching for glycans in the PDB. Because if you've ever tried to do this, you realize that it's an extraordinarily difficult thing to do. The protein database was created to store proteins, and no attention was given to how to store carbohydrates. And this created problems because, for example, there's only one way to talk about alanine, it's alanine. But glucose, oh, that could be a different thing, because if you say alanine plus alanine plus alanine, you get a tripeptide. But glucose plus glucose plus glucose doesn't give you one molecule, because they could be all linked in different ways. The PDB used to let people name that trisaccharide with a name instead of calling it as being made up of residues. So the whole thing became a nightmare. So as of yesterday, there were 162,000 biological macromolecular structures in the PDB. And this is not just proteins or glycoproteins. It includes DNA, RNA, lots of stuff now. But it's been impossible to say how many structures contained carbohydrates. There were estimates of about 10%. Now, using the tools we've developed, we now know there's around about 20% of all the protein structures in the database that also contain carbohydrates. The problem with having the lack of tools to do this is it means that there's lots of erroneous structures in the PDB. Things where the structure was deposited by a crystallographer who has expertise in protein structure, but not expertise in carbohydrate structure. And without the tools to check and warn them that the structure is wrong, it just gets deposited. And this leads to a lot of confusion. And it leads to propagation of errors when crystallographers retrieve a structure and intend to use it for molecular replacement to solve their next structure. They reuse or they download a structure with an error. And this really reduces the utility of the data. Data is very valuable in other ways. So we took on this challenge. If you go to this GlyFinder, dev.glycam.org/gf, you come up to this site. So let's just see if it works as we hold our breath. I hope everybody can now see a new interface. And what we're going to do is we're going to search for some glycans in the PDB. And as you notice, right, I hope you can see this in the search box, there's a star. This means it's a wild card, and you'll see why in a minute. But let's search for N-acetylglucosamine that's beta-linked to asparagine. And the glycobiologists in the community will go, okay, I understand this is the start of an N-linked glycan. And now you can see the point of the star. Because this could be a much bigger structure, it includes everything to the left of the glycan there. All right, so you click done and it does its search. And that's how fast it is. It retrieved 47,000 glycans, or actually glycoproteins that are connected to asparagine. Let's pick one of these, and this is shown in SNFG representation. If we go here, it allows the user to visualize it, right? And you can scale and interrogate the structure. And we've incorporated the 3D SNFG motifs so you can rapidly see where the sugar is and what the sugar is. Alternatively, you can click the More button and get some details such as what's the resolution of the structure, the mean B factor of the structure, the oligosaccharide B factor. These are quality assessments. You could download the coordinates of the PDB complex or of the sugar or the fastest sequence. And of course, you can see the structure. So on the left here, it lets you filter. For example, we might want to say, well, I just want high resolution structures. So let's do 2.5 angstrom resolution. And if we do that, it took 47,000 down to 9,178 structures, right? So, and it got rid of a lot of spurious, well, low resolution data. Okay, so that's what I wanted to show you about that tool. If we go back to the presentation now, this just summarizes the same thing. Bits and pieces we've learned. Well, this was a little old, so it's not quite up to date, but if we think of N-linked glycans, there are 5,000 PDBs that contain N-linked glycans. There are 25,000 N-linked glycans among those 5,000 PDBs. So on average, about 5 N-linked glycans per glycoprotein. Does that matter? Not particularly, it's just a little factoid, but it's the sort of thing you can mine out of this. Other information, O-linked glycans, well, they're represented at a much lower level. Okay. So moving on and getting to what I really want to talk about in the main part of the talk today, which is the COVID work. What we're asking now is the question, how does glycosylation affect my protein? Well, let's think about how we have to model a glycoprotein. First of all, we need a protein structure. And then we have to decide where are you going to put the glycans? So at what position, for instance, asparagine 88, asparagine 187, or so forth. And what glycans you want to put at each site. So perhaps, and this is a work in progress. So when you go there, it won't look like this, but this is the philosophy. You might choose, I want all high mannose, or you might choose something else, or you might create a custom structure. Then we have to add them to the structure. And so the first thing it does is it builds a 3D structure of the sugar, translates it to the protein surface, and then resolves overlaps. Well, what does that mean? Let's look here. Here's a erythropoietin. It's got 3 end-linked glycosylation sites, well-known crystal structure. We want to put a sugar on it, on each site. Okay, so we do that. Now, right away, if you look at this, you can see, wait a minute, this glycan looks like it's bumping into the protein. Why would you put it there? Well, I wouldn't if I could see it, but the computer can't see it. So all it does is it attaches it, and then we have to figure out, is there an overlap? And we have to move the sugar out of the way. If we keep all of the atoms in the calculation, then there's almost 500,000 calculations that have to be done every time you want to move the sugar to see whether each atom is bumping into any other atom. So we simplify this by making amino acids not based of atoms, but based of spheres with a backbone sphere and a side chain sphere. And we simplify the sugars by making them beads as well. All right, and this makes it quite a bit faster. So that lets us now say, okay, we detect an overlap and we can move the glycans and resolve it. Okay, but we know that sugars are flexible. So is one single shape representative? And that's certainly one question. If we look at the structure, there could be many, many shapes, orientations of the sugar that could be equally likely or at least acceptable. So one of the questions we have to ask, beyond just simply what is a static shape, is how do you get a model that takes into account the motion of the sugars? And secondly, when you've got something like GP120 here on the right, which many of us know is a very heavily glycosylated protein, how do you resolve these overlaps? Because you've got such a dense packing of sugars, they bump into each other, and it's an extremely complicated algorithm to figure out what nature does so simply. And the way we do that is we use molecular dynamics. And here's an illustration of a glycoprotein. This one happens to be from hemagglutinin, from influenza, which is heavily glycosylated. And you can see during the simulation, the protein is wiggling and the sugars are wiggling. And if you run this for long enough, what you get is a realistic model in terms of how much motion the sugars have, how many shapes they adopt, and so forth. So this is essentially the gold standard for modeling glycoproteins. The question becomes, how long do you have to run the simulation in order to convince yourself that you've sampled all the shapes? But this is really where it stands. So given that we're able to do this, and here's our GP120 model spinning around, what can this help us do? And from the very simple perspective, if you've looked at a biochemistry textbook, you often see proteins with at least a primary sequence, get a little Y or something like that in the sequence to indicate a sugar. Many people have no concept of how big the sugar is relative to the protein. The 3D SNFG is sized, appropriate to scale, and so you get right away just a simple, is it likely that the sugar is important? And you just get a visual for that. You can also look at, for example, there might be a site down in here that isn't glycosylated, and if you just look at the sequence, you don't know why it isn't glycosylated. But when you look at the 3D structure and you think of the size of the sugar, you can determine that it is inaccessible. Further things, you might say, well, why is there a patch here that's all high mannose? Why isn't it processed? And that's because ERMAN1, the enzyme that would normally process or begin the processing sequence of events, can't physically access it without bumping into the underlying protein. And so you can predict processing. And then lastly, just as you're looking at this, you can see, well, some parts of the protein seem to be shielded visually by the sugars. Well, this visual shielding, and this is what we're going to talk about for the rest of the talk, is what an antibody faces. If it's an antibody that is trying to recognize the protein, if there's a sugar in the way, literally the antibody cannot bind to it. And so this is called antigenicity, the interaction of an antibody with an antigen. And so we would like to be able to quantify that. And I'll illustrate this with the SARS-CoV-2 spike protein. But that leads us now into just a discussion. And, you know, I think everybody in the world is becoming a virologist right now. So correct me if I get this wrong, but this is a picture I got online from a blog, and it summarizes the basic view that many people have. Here's our virus, and the spikes on the outside are the proteins that grab onto the host receptors. And in SARS, coronavirus, it's acetylcholinesterase, and this is what it grabs onto. And there's a protein here that's a protease that's also important for infection. So there's a lot of interest in this initial interaction. Then the virus replicates and it buds out. And we're going to talk about a couple of features. One is this, and how glycosylation affects that. And one is this bit here, Golgi ER, and how glycosylation is important there. So if we zoom in on this image now, this spike protein here that comes out of the SARS coronavirus actually looks like this. This is a structure that's based on an electron micrograph, a cryo-EM structure. And the cryo-EM structure is good, but it doesn't have all the loops resolved. And so the loops were modeled in, and this is obtainable from the Swiss model server. The coloring in red, if you just look at it, you'll say, oh, that seems to be on the outside, and black is on the inside. And so red is the accessible bits, and it is. It's accessible to antibodies, and we'll talk about how we've computed antibody accessibility. This is the non-glycosylated spike protein. The receptor binding site is up here at the top at the apex of the S1 domain. The protease cleavage site between the S1 and S2 domains is down here. And this is really just to orient you into the discussion. But now let's look here. Once the virus is endocytosed and you have replication going on and it's being assembled, the protein passes through the host, Golgi, and endoplasmic reticulum and buds out. Now, everybody in this audience probably knows that one of the key purposes of the Golgi and ER is that it adds glycans onto this spike protein and onto envelope proteins of viruses in general. That's, of course, not shown here because this is a generic image and glycans apparently don't make the cut. But they are important, and I'll show you why. Reviewing your basic glycobiology. In the ER, in humans, N-link glycans are attached from a dolichol phosphate precursor as this preformed Man9 glucose 3 glycoNet 2 structure. As they go through the ER, the glucoses are trimmed off. As they enter the Golgi, they get processed more and more and can have a staggering array of modifications. But here you can see, for example, a complex type or sialated with sialic acids and so forth, right? So this is what's going on, and this depends on the type of cell. And that's really going to be quite critical for what we're going to be discussing. In terms of influenza, because there's a large body of data on this and it's been very well characterized, any one site can have many different glycoforms, because if we go back to this, each of these enzyme processes doesn't go to 100% completion. So at this one site, some of the forms are going to have this, some of them will have that, some of them will have this. So you're getting potentially a mixture of possible glycoforms. And from the glycomics analysis, this is how they're annotated. You can see it's small sugars to the core glucanacs that we searched for in GlyFinder with trimannose, typically something like that. So this is posi-mannose type structures, plus or minus fucose, okay? That's one thing we have to consider for the modeling. What glycan are we going to put on each site? And the second thing, of course, is that where the glycans are makes a difference. Now, when we look at influenza, again, stay with this very nice paper that looked at expressing hemagglutinin, or rather expressing virus in cell lines that are typically used to produce vaccines that target influenza. You can see in SF plus cells, or high 5 cells, or HEK cells, or eggs, you get a huge difference in the types of glycans that go on each site. This is just one site, right? Just site number 1. It has all these different possible glycoforms detected. And just to give you some idea of the variation that's shown on the bottom here, right? So from a modeling perspective, when a glycomics person publishes this data, it's very difficult to decide what to do with it. So what we did was we took the most abundant glycoform, glycan at each site, and went forward in our modeling with that. The other question we want to think about is, as viruses circulate, they undergo antigenic drift, leading to seasonal variations in the virus, or antigenic shift as different viruses combine, and different strains combine, and you can get pandemics. And often these can lead to the creation of new or deletion of old glycosites. And when you do this, you're changing the parts of the protein that are shielded by antibodies, shielded by sugars from antibodies. And this means we've got a question. Can we predict the impact of glycosylation on protein antigenicity in anticipation of antigenic drift or shift? Right? Because if we're always working after the fact, then we can't do anything to anticipate creation of a vaccine. Now switching fully on to COVID, here's the structure of the non-glycosylated spike protein, and here's an overlay of structures that we generated using MD simulations. The different types of glycans we chose came from a group from Max Crispin's group, and it's glycans that were observed in HEK 293 cells. These are human embryonic kidney cells. Just looking at it, you can see, well, we've got some kinds of glycans here, different ones in pink, a high mannose one here. Now, this one, you could say, why is it high mannose? Well, it's tucked in so deeply, it can't be processed further, okay? Other conclusions in general, well, here's the receptor binding domain up here. It's not glycosylated heavily. And we have the fusion domain. The cleavage site down here is not glycosylated. And these are for functional reasons. We'll get to that in a minute. So with this, what can we do? Well, we can look at it from different angles. Here's the top view, and there's the receptor binding domain, and you can see it's unhindered by sugars and therefore able to interact with the ACE2 receptor. And side view, and there's the receptor binding site. Okay. But we would like to see how this relates to antibodies. So the first thing we did was we took a single structure from this, and there are several co-complexes from SARS coronavirus 1 and the MERS coronavirus that have been co-crystallized with known antibodies. So that we aligned the SARS and MERS spike protein on the SARS protein from SARS-CoV-2 just to see where the antibodies might be located, right? Where the epitopes were that they might target. But now we're working with SARS-CoV-2, and it's got different glycosylation, and there are several glycans that would in principle collide with these antibodies, and therefore the antibody can't bind there. So that eliminates them, and it really leads to the idea that there's an epitope up at the top here, the receptor binding domain, where antibodies are likely to find some traction and find useful epitopes. But that doesn't mean other regions are not accessible, because there are countless antibodies raised when you get an immune response, but only a handful have been co-crystallized. So what we really need to do is quantify how much of the surface is actually accessible for antibody binding. The way we do this is we want to probe the surface with a simple model of an antibody. And the simplest model we could come up with was to use a ball the size of a hypervariable loop in an antibody. So first of all, we worked that out, an average size ball. And we roll that over the glycoprotein surface, and we see when that ball can touch the protein. That would be when a hypervariable loop can make contact. But we also have a requirement that the hypervariable loop has to be close to the variable framework region of the antibody. So there's a second criterion here, but this is basically our model for what an antibody looks like, and we use it to quantify how much of the surface of the spike protein can be contacted by an antibody. And we did this as a function of different glycans. Now, the models you've seen previously were this HEK site-specific glycosylation. And it said that 41% of the surface was shielded from recognition by an antibody, according to our model, whereas 0% of the non-glycosylated surface was shielded, right? Because there were no glycans. These other glycans, well, when we first started this project, there were no glycomics data available. So we didn't know what glycan to put at each site. So instead, we thought, well, we'll use one that corresponds to something that we might see in influenza, depending on the cell type. That's these three. And we will use high mannose because that's like before it enters the Golgi. And we'll just see what we get. Now the smallest, the smallest glycan shields the smallest surface, only 30%. Not a big surprise there. What is interesting, to me at least, is that each of these other glycans arbitrarily shield about 40–45%, which is more or less what we get from the detailed analysis of the glycomics. So this is significant because it means that if you want to model glycosylation, or at least the shielding of a protein surface by a glycan, as long as you choose a reasonable sized glycan, you're going to get an answer that's reasonably correct. If you care about this 5 or 6% difference, then of course that isn't the case. It might not suit. But if you just want to know what regions and approximately how much is shielded, it means you can go ahead and make some models without knowing the exact glycans. And what we did with this was we then mapped the surface exposure onto the protein. So what I've got here is the sequence of one of the protomers. It's a trimer, so it's one monomer. And colored in red is the accessibility of that amino acid to an antibody. And let's look first at this residues reported to interact with the ACE2 receptor. Okay, so these are residues that underlined in blue that form the receptor binding domain. And you can see that they're all really highly accessible, which is exactly what we saw in the 3D structure. You can also see that there are no glycans. How do we know that? Well, the glycans are shown with an asterisk above the relevant asparagine. So there's no glycosylation. Coming down here, this is the protease cleavage site. It's also really quite exposed and not glycosylated. Other things we can look at, in the boxes, what I've done is I've looked at the peptide sequences that are predicted to bind to MHC2 or human leukocyte antigens, which are relevant to raising an immune response. So the ones in boxes talk about immunogenicity, not antigenicity, and therefore they're relevant to vaccines. And so by looking through this data, and we've got a project where we're working on picking out peptides based on this to use as immunogens, you can say, well, is the glycan important or not important for blocking potentially the binding to an adaptive, an HLA, which is going to be important for adaptive immune response. All right, so to conclude, sorry for all the text, but glycans shield about 40% of the surface. really independent of the glycan as long as it's a typical kind of glycan. This suggests that these models can be used in advance of the reported glycomics data, which means you can model the effects of glycoside alterations in anticipation of antigenic drift. This is really quite an important feature, because if we're always being held back because we have to get good glycomics data, then, well, this is saying you don't have to be. You might need that data if you want really to explore specific interactions. Other things we've learned that are perhaps obvious is that the most accessible and largest epitope consists of the ACE2 receptor binding domain. And this is fortunate because the virus can't make mutations there without affecting its ability to infect. It can't glycosylate there, or it can't bind to the host. So as long as the coronavirus keeps wanting to bind a large ACE2 receptor, then this is likely to be a good epitope for a vaccine. And that's indeed what we see in the antibody co-complexes in the MERS and SARS structures. Whoops. Okay. Now, when you look at different glycans at each site, this is called microheterogeneity. What we're looking at there, we've said it doesn't really matter too much in terms of how much they will shield the surface, but it will matter a lot for how the innate immune system reacts to the virus, because the innate immune system uses lectins to grab onto those sugars as a way of neutralizing the pathogen. So this is really quite important, because depending on the cells you saw, you get different microheterogeneity, you get different glycans at each site. Each of those constructs will have different innate immune responses. And you may say, well, who cares, because those are all artificial, they're not human. But microheterogeneity varies between individuals. Gordon Lauk has shown this in several publications. So it depends on many factors, including age, underlying disease, ethnicity. And as you hear these factors and you think about COVID-19 pathogenesis, to me at least, I start to think that glycocyte microheterogeneity may very well be responsible for differences in innate immune response between individuals. So looking at these 3D structures may really help in when we get glycomics data from isolates, we may be able to relate that to pathogenicity. And anybody who wants these structures, you're welcome to download them. Go to www.glycam.org. And just to thank the relevant people, Oliver Grant is the creator of the glycoprotein builder that we used and did all the SARS modeling. Keigo Ito came up with the antibody accessibility probe that we used to quantify accessibility. Dave Montgomery has led the creation of the GlyFinder tool. We have a lot of background work going on in making the website and all the tools function, and that's driven by Lachelle Foley. And Dan Wentworth has played a big role in the architecture of the new site and in making it user-friendly. So lastly, thank you guys for the opportunity to give a talk here, and to the NIH for support.
Thank you so much, Rob, for an amazing talk about a very timely topic, and it's good to see you.
All right, thanks everybody.